Leading a startup team? Whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Glioblastoma is a devastating and fatal brain cancer that progresses rapidly. Median time from diagnosis to death is 12 to 15 months. In recurrent cases, treatment consists of both symptomatic and palliative therapies, but the disease remains fatal. VBL Therapeutics is developing a targeted anti-cancer gene-based therapy that is in late-stage testing for recurrent glioblastoma. We spoke to Dror Harats, CEO of VBL, about the therapy, how it works, and why it might be useful in treating a variety of cancers. Dror, thanks for joining us.
Thank you, Daniel.
We're going to talk about an aggressive form of brain cancer known as glioblastoma, multiform VBL Therapeutics novel experimental therapy that's in late-stage development to treat it, and its potential to treat a broader range of cancers. Let's start with the disease itself, though. What is glioblastoma? How common is it? And how does it progress?
So glioblastoma is the most aggressive malignant brain tumor, and actually it's the most common malignant brain tumor. And although it's an orphan disease, yet it's actually been diagnosed in the United States alone in about 12,000 patients every year. And usually it happens in midlife, in men more than in women. So it happens in the prime time of people in their life. And it's quite a devastating disease because from the time of diagnosis until the time that the patient actually die, and usually most, if not all patients will die from the disease, it's actually only as a median 15 months. So it's really one of the most devastating types of cancer. And at the time of diagnosis, usually the patient get a surgery to try to resect as much of the tumor as possible. Not everybody can get it resected, but at least that's the aim in the beginning. And then usually the patient will get a combination of radiation therapy and chemotherapy with Stimodar. So with this triple therapy of surgery, radiation therapy, and Stimodar, unfortunately, half of the patients will have a recurrent of the disease within 6 months, which is quite terrible. And from the time that the disease comes back, actually we don't have really good treatment for that condition of recurrent GBM. We do give sometimes more surgery, more radiation therapy. People give Avastin, but none of this treatment actually showed a statistical significant increase in overall survival. So without any treatment, the median time of to death between the recurrent is about 6 months. With all the new therapies, it can get to 7, 8, sometimes 9 months, but that's the most that we can achieve. So there is a major need for treatment for this disease.
Well, there's been some effort to use anti-angiogenic treatments such as Avastin, which are intended to choke off the blood supply of tumors and kill them. How are these delivered, and have they provided any benefit for glioblastoma patients?
Actually, one of the major things in a brain tumor that it actually happens in a closed box of the skull, and there is what we call a blood-brain barrier, which actually should protect our brain prevent a lot of drugs to get to the brain. Anti-angiogenic therapy has been developed for brain tumor, and the most common one, of course, is Avastin. And it's actually been given to patients through an IV infusion, and it's getting to the brain. The only major issue with it is that it works by shrinking the blood vessels and actually decreasing a little bit the swelling in the brain, but it has no effect on the tumor itself, and therefore we couldn't show, or the company that developed it actually couldn't show any benefit in terms of increase in survival. The only thing that they could show that there are some responders because they reduce the fluid around the tumor. And now you can ask yourself a question, why does the FDA allow this treatment? So in the beginning they allow it because of the some response rate and because there is nothing else, and then it was conditionally approved. So the company had to show that it actually really increased survival, and two trials actually failed to show increased survival, but there is nothing else, and therefore it's still on the market. Having said that, at least with our drug VB111, which we are developing already in a phase 2 trial that we were doing, we could show that we almost double the survival of the patient with recurrent GBM for about 8 months to 15 months. And that's really encouraged us, with the support, of course, of the agency, to go ahead and do a single trial to show survival, which is a trial that we are running right now, the GLOBE trial. And it was agreed with the FDA under an SPA that when we will show survival benefit in this trial, the drug is going to go on the market. And hopefully that will happen quite soon, because we are expecting results in the beginning of 2018. And if indeed we are showing a significant benefit in survival, that will be the first drug in recurrent GBM.
Well, let's take a step back. What exactly is VB111? It uses a viral vector to deliver a gene. How is it delivered, and what's actually being delivered?
Okay, so that's a very good question. VB111 to the doctor and to the patient is a simple IV drug. The patient coming to the clinic can get an IV infusion like many other drugs for cancer for about half an hour and then go home. But this is a very sophisticated drug, because what we are delivering is actually a therapeutic gene that's going to work very specifically in the place and the time that we want it to work. And the way it's actually built is we are using what nature is using to deliver genes. That's basically viruses, because viruses know how to deliver their own genes into cells. So we are using a common flu virus, an adenovirus, that does not know how to replicate itself anymore, so it cannot cause any disease anymore. It's just a way to deliver our therapeutic gene. Our therapeutic gene is actually a deadly gene that's known to kill cells that it expresses. But the uniqueness about this therapy that every gene in our body has an engine for the gene that tells the gene where and when the gene should be expressed. And we have a unique technology with a piece of DNA that actually tells this deadly to express itself only when there is new building blood vessels. And usually in our body we don't build new blood vessels unless we have a tumor. So the way this drug is working is actually we are giving the drug all over the body, and it goes all over the body. But only at a place where we have new blood vessels, it's going to express itself and actually destroy the blood vessels. So one can ask itself, so what's the difference between this and, for example, other anti-angiogenic drugs like Avastin? So the problem is that tumors has many ways to build new blood vessels. And Avastin just works on one of them. And then, of course, the tumor can use many different ways. But with our drug, we don't care how the tumor is building its new blood vessels. The moment that there is new blood vessels, the deadly gene will actually destroy them and kill them. More than this, we are not normalizing in any way the blood vessels. We just destroy them, and therefore that affects the tumor directly. But if that would have been the only mechanism, that's just half of the work. The other thing that we are doing is actually educating the immune system to fight the tumor. And the way it has been done, it's what we call now, it's a new field in medicine, what we call viral immuno-oncology. Our whole immune system is geared to fight viruses and bacteria. That's the way the immune system is built. Now, even when you have a tumor and you shut off the immune system, if there is a virus there, the immune system will find its way to get there. And because we are using a virus and we are actually changing the tumor and the blood vessels, all of a sudden the immune system recognizes the virus and of course it recognizes the tumor, and therefore we're getting an immune effect of the tumor. So because we are working in two major mechanisms on how tumors are actually surviving, one is building new blood vessels, we are not allowing, and the second is avoiding the immune system, and we are bringing the immune system to kill the tumor, the drug is very efficient.
As I understand it, this is not a one-time administration, but something that's infused regularly. Given that you're using a viral vector, is there any issue with creating an immune response that renders the drug ineffective over time?
So you're completely right, because we are not trying to put any new gene and you just give a one-time treatment. It's every 8 weeks, and every 8 weeks we're coming with a lot of the viruses, and we infuse it into the body. So even if you have a lot of antibodies against the virus managed to get in enough amount to the right place in the tumor. But more than this, the more immune reaction we get against the tumor, against the virus, the better immune reaction we get against the tumor. So the patients that develop high temperature, for example, when we infuse a virus, or patients that get all kinds of cytokines in response to the virus, is actually the patients that benefit more from the treatment. So this immune reaction against the virus is actually very helpful to the patient and to the drug to actually do its work, and therefore we don't care that the immune system is fighting the virus. We actually want to fight the virus.
And is there any issue getting that across the blood-brain barrier?
Not really, because this virus know how to infect the brain, so we have no issue at all. And actually, because we are destroying the blood vessels within the tumor, we're actually destroying the blood-brain barrier, and we are allowing the more and more viruses to get there. So it's actually working completely opposite to Avastin, because Avastin is actually normalizing the blood vessels and basically normalizing the blood-brain barrier, and therefore, when patients fail Avastin, usually they don't respond to any other drug, because basically other drugs cannot get into the brain. It's not the case with the VB111, because we are actually enabling more fluid and drugs to cross the blood-brain barrier. So, for example, when tumors grow after Avastin, they grow in a way that you don't see the fluid, you just see the tumor grows into the brain. With VB111, you do see a lot of fluid and much less tumor. And of course, the fluid then goes away, and the tumor is much smaller.
Targeting newly formed blood vessels is a good thing when you're going after tumors, not so with other tissue in the body. How selective is VB111?
So VB111 is extremely a selective drug. Otherwise, we wouldn't be able to give such a toxic gene or a deadly gene into the body and don't get really side effects from the transgene. And the reason is that in normal adult life, we don't build new blood vessels. So there are only very small certain conditions that we build really new blood vessels. Even in the times that we don't have enough blood supply, like in a heart attack or in a surgery, we don't really build the same type of blood vessels that we do build in cancer, and therefore the drug is not expressed there. Just as an example, we had 2 patients that needed to get an operation very quickly after getting the drug for another reason, of course, not because of the drug, and both of them recovered completely well. And we didn't get to any major side effect regarding other blood vessels in the body because they are not the type of new building blood vessels that you do see in tumors. So it's extremely specific, and actually in the blood we find the virus, of course, but we cannot find any evidence of the new gene or the transgene, the deadly gene, because it's not expressed in the body. Only when we had a chance to do a biopsy from the tumor, we could find that the gene is expressed in a very high level. So it's expressed only in tumors.
And you spoke to this earlier, but what do we know from the about the efficacy of the therapy from clinical trials to date?
So we already were doing quite significant clinical trials in human beings, and actually by now I can say that about 400 patients already got this drug so far in a couple of phase 1 and actually 3 different phase 2 trials. And these phase 2 trials were done both in recurrent GBM, in ovarian cancer, and in thyroid cancer. And what we learned that it extends survival in all indications. So in GBM, which is what we are talking about today, recurrent GBM, as I said before, it's actually almost doubles the survival from 8 months to 15 months. We also noted that it actually blocks the growth of the tumor, and that's why increase the survival, because if the tumor has no blood supply and the immune system is fighting the tumor cannot really grow. But we are developing for recurrent GBM for a brain tumor, but it's a drug that by its mechanism on the immune system and on the blood vessels should work in other indications. And indeed, in patients with a late stage ovarian cancer, which is the ladies that don't respond anymore to chemotherapy or are actually refractory to chemotherapy, we could also show that we increase the survival from about 6 months to more than 800 days, and there we could show a significant response in biomarkers and in other modalities. Same thing with thyroid cancer. And in all these indications, we do get a very nice tail in the curve, or what we call that a significant percent of the patients are actually living for a very long period of time. So just as an example, in this thyroid cancer, where patients were actually failed every other therapy, we do have a group of patients, which is about 30% of the patients that now are alive for almost 4 years and actually don't die. And the same thing we could see in some patients in the ovarian and in some patients in a recurrent GBM. We have a patient that actually responds to BB111, our drug, with a complete response, has no evidence of disease now for more than 3.5 years.
You've got both orphan drug designation and fast track designation in the U.S. and Europe. You mentioned you're doing this under a special protocol assessment. All goes well, how soon might you be able to take this to the market?
So actually we are expecting the results in the first quarter of 2018, and of course because it's an event-driven trial, which means that certain events has to happen, and only when they happen we are closing the trial, and the longer it takes, it actually tells us patients live longer. So we expect it in the first quarter of 2018, but if it will take a bit longer, that will be a very good sign, because that means that more patients are alive for a longer time. And I believe that from that point, and including submission of the whole BLA, we could see the drug sometimes in 2019 on the market.
And is the expectation you'd commercialize this on your own, or would you seek a partner?
Actually, it's an orphan disease, and we could definitely commercialize it ourselves for recurrent GBM, but this drug has a major potential in many other solid tumors. So we are going to be a little bit opportunistic here, selling it ourselves, but also looking for a strategic partner to help us develop it to many more different indications, because it's not going to be just ovarian and thyroid and GBM. We already know from the phase 1 that there is a good chance it will work in lung cancer, in renal cancer, in neuroendocrine. We actually plan to start study toward the end of this year or beginning of next year already in lung cancer in combination with one of the checkpoint inhibitors. So we believe that the potential here is really huge, and therefore we can start selling it for the first indication ourselves, but eventually it would be good to have a strategic partner along the way.
Dror Harats, CEO of VBL Therapeutics. Dror, thanks so much for your time today.
Thank you very much, Daniel, for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.